Yeah, that looks good.
Good. Okay. Thank you, TK.
Great. Yeah. Cool.
Yeah, actually, I just got the acceptance letter this morning for this paper.
Oh, great. Good timing.
Yeah, good timing.
We always like to have the cutting edge things on NeuroZoom.
Just keep it like that, waiting.
Maybe stop sharing it. I just have to show like a introductory thing, and then you could share it again.
Okay.
How's Hangzhou?
Yeah, sunny.
Nice. Yeah, I'm back in Shanghai, but I was in Xishuangbanna for a couple weeks.
Yeah, I saw you, 朋友圈.
Oh yeah. We were heading to the airport, and then like there was like a single case in like a city and nearby, so they locked us all down, and we couldn't leave, and we just had to stay a while longer. But then we got out.
Hello.
Hi, Jianqin.
Yeah, hi, Erin. Hi, Yihui. Can you hear me?
Yeah.
Yeah, thanks for coming. Do you want to test your slides out, Jianqin?
Yeah. Let me.
Thanks.
I didn't find the button to share the screen.
There should be a green button on the bottom with an arrow pointing up that says share screen.
Yeah, I found it right now. Can you see my screen?
Yep, it looks good.
Let me roll down the whole thing. Yeah, I think it works well.
Yeah, looks good.
Okay.
Yeah, great. How's Chongqing doing?
Super cold.
Yeah, what's super cold? It's super cold in Shanghai.
Also super cold in Shanghai.
I think so.
Oh, I see your WeChat. I thought you are in Yunnan province.
Yeah, I was there. I just came back. I came back last week, but I was there for a couple weeks in Xishuangbanna. It was very nice there.
It looks warm. Where
Yeah,
is
very warm.
Sure.
Yeah, very warm. So when did you come to Chongqing?
When?
Yeah,
I-
or when did you move back? Yeah, when did you move back?
2017.
Okay.
I was always in Chongqing. I got the PhD here, college here.
Mhm.
Just moved to San Francisco for postdoc, and then moved back.
Mhm. 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 Yeah, my a postdoc in my lab. Her parents are in Chongqing, and yeah. So when I was out in Chongqing, I visited them.
Quite nice city. It's not too big, but have very like delicious food and views. And I think
Yeah.
sometimes it really looks like the San Francisco, the real,
Yeah, with the
real
hills and
downtown.
the
Yeah.
yeah. Yeah, yeah.
So Yihui will be the first. Yeah, right?
Yeah. Yes. Yes.
Okay. Okay. So do I need to turn on the video all the time, or I should mute and turn off the video until?
You can it's up to you. You can mute your microphone, but you can keep your camera off or on, whatever you feel comfortable
Okay. Okay.
with. Yeah, thanks for presenting your work. We're looking forward to hearing it.
Okay.
And thanks for coming early just to check things out. Sometimes there's some issues with the slides, so it's just good to make sure it's okay.
Mm-hmm.
So Yihui, is your lab in the same campus as Hailan's?
Yeah. Yes.
Good.
We're very close in the fourth floor, but we are
Oh.
separate.
Mhm, mhm. Yeah, I'll try to come back to Hangzhou maybe sometime in the next couple months.
Welcome.
Thanks.
Aaron.
Hi,
Hey,
Zilong.
Zilong.
Great. Nice to see you.
Nice to see you.
So long. I saw something on the news about a monkeys running around the streets of Shanghai. I don't know if that's from your lab
Hi?
or what.
No. So maybe some Zoom, right? Run out Zoom. I don't know. Is that
Okay.
oh, that's kind of scary. Maybe
It
not.
was like in Xiaoyang.
Oh, is Jianqin here yet? I'm asking.
He was here.
I'm
He
here.
tested his slides.
Is okay. I'm trying to find. Great.
It was quite an ordeal to get back to Shanghai.
Oh no. Is it?
So
They don't have flight come back or?
no, well, because we were there for 8 days and we're about to head to the airport, and
Right.
then they said, so actually before we left, I gave a talk at Tongji University and I had dinner with Yisan, and
Yeah.
she said, "Oh, I know someone in the Sichuanbana government in case you have any trouble," and I said, I'm not gonna have any trouble. Don't worry about it." He's like, "No, just
Okay.
take this." So then we're leaving, and then there was like some positive case in Kunming or something, and then they locked down
Oh,
all of the city we were in,
yeah.
and we couldn't get to the airport. They canceled all the flights, and
Ah.
then we took COVID tests, and then they said you can leave if you take 2 tests within 48 hours, and then get like a stamp, government stamp. But they made it so you can't do all those 3 things in 48 hours because they stopped the testing, and so we just
Oh.
kept anyway.
Yeah.
We were then I was like, oh, then I remembered Yi's son mentioned that. So then we
Yeah. It's
got some help, got the tests, got all completed. Then we took a train to Kunming, and then
ah.
Kunming we flew to Shanghai.
Oh, it took a while.
Yeah,
It's
but
good to have some connections. Yeah.
yeah,
People just don't know what to do, right? For governmental stuff, they just no idea what to deal with the tourists going around.
yeah, yeah. So it was tricky, but it's if you have to pick one place to be locked down, this is a nice
Oh,
place.
yeah. It's how the weather is. It's not too warm, right?
It was not hot, but it was warm and nice,
Okay.
and it was good. Yeah,
It's
yeah,
better
it was
than
really
Sanya.
beautiful.
Yes.
Yeah.
Sweet.
Did you go anywhere?
No, just local. Just I'm afraid I cannot go back as if something turned out. So
Yeah. I have to go to Beijing next week for a little bit, but that should be okay.
yeah, but it should be fine.
Yeah. I got a star from going to Kunming just for a few hours,
Okay. Oh.
but it should be
Okay.
okay.
Oh, you've been there, so
But
yeah. Okay.
yeah, should be okay.
Shanghai is clear finally. So
Yeah.
yeah. Yeah, we hopefully expect some coaching power Suzhou meeting this year. We're planning for the autism meeting
Oh,
in
great.
September 2022. So hopefully it's make out in some way. Yeah.
Yeah, I hope so.
I don't know. Mhm. Nice. First new Zoom.
Great. Okay, maybe you should get started.
Sure.
Okay.
Yeah.
Hi, everyone. Happy New Year. Welcome to a new season of NeuroZoom. And we have awesome talks starting today. And as usual, I want to let you know about next week's talks. We have Michelle Manji from Stanford and Chunghua Gu from Harvard giving talks. Don't miss those. And now it's my great pleasure to introduce the first speaker of the year for NeuroZoom, Dr. Yihui Cui. Yihui is an investigator at Zhejiang University, and she earned her bachelor's degree at Ningbo University, then did a master's at East China Normal University before going to Paris for a PhD, where she investigated the role of endocannabinoids and spike timing. From there, she moved to do a postdoctoral fellowship with Hailan Hu, former NeuroZoom speaker and friend of NeuroZoom at Zhejiang University. And here, Yihui made a major impact, and she started studying the lateral habenula. This is the part of the brain that's associated with negative emotions. And she had back-to-back papers in Nature, one of which she's the first author, and the other one she's a co-first author. And there, she and her colleagues described in both of those papers the role of burst firing in the lateral habenula neurons using a rat model of depression. Then they showed that the drug ketamine blocks this burst activity, and then they even go on using a proteomic screen to identify the target, which is surprisingly an astroglial potassium channel. And then they show that the levels of that channel vary based on activity as well as depression behaviors. So their work explains how ketamine works, explains and presents a new drug target. So those exciting findings have catapulted Yihui to her own laboratory, and she's going to describe her exciting new findings with us today. Take it away, Yihui.
Okay, thank you. Can you see my screen? PowerPoint?
Yes.
Okay. Okay. So thank you, Aaron and Zilong. It's my great pleasure to share this unpublished work here. Actually, this paper just got accepted in Neuro several hours ago. And my talk is about a habenula-centric neurocircuitry mechanism of chronic stress-induced depression. So in the beginning of my talk, I would like to mention this black dog, which Sir Winston Churchill once used to describe the dark mood period that he suffered, the major depressive disorder. Actually, 1 out of every 5 people has experienced this sometimes in their lives, and nearly 1 million commit suicide every year. So for decades, researchers have tried to find the neuromechanism of depression, thus to tame this black dog. However, numerous studies have focused on the cure, but less on the prevention. In other words, people paid much more attention on the neurodysfunction under depression state, but less on the dynamic change of neurocircuit before depression onset. So among the complicated etiologies of depression, chronic stress is possibly the biggest black dog. My postdoc work with Hailan Hu, we know she is a great scientist working for 10 years in the depression field, and we decoded habenula burst is an electrophy marker of depression status. So after I finished my postdoc with her, I shifted my research field to dynamic neuromechanism of chronic stress before depression onset. The goal is to find early intervention strategies to prevent depression. And among various neurocircuits involved, we have been focused on the habenula-centric neurocircuit. The lateral habenula, LHb, has been recently implicated as a convergent hub that integrates value, sensory, and experience-dependent information to mediate various motivational processes and the pathophysiology of depression. And circuitry-wise, LHb acts as a gateway from the limbic forebrain to midbrain aminergic nucleus. It is shown to inhibit the monoaminergic reward center via GABAergic RMTG nucleus. And my postdoc work with Hailan Hu discovered enhanced bursting in LHb encodes depression status. It generates a stronger output to inhibit the downstream aminergic centers and thus cause depression. But however, LHb is a subcortical brain region and which cannot directly detect and process stress information. It remains elusive whether and how its upstream afferents physiologically involved in processing stress experience and how prolonged the stress exposure gradually scoops LHb-centric circuitries and finally elicit depression. So we first used CFOS as a proxy of neuroactivity under stress and labeled LHb projecting neurons by injecting a retrograde tracer, CTB, into the LHb. After 1 week, 1 week after CTB injection, we tested the mouse with various stressors, including restraint stress, social defeat stress, and food shock. CFOS and CTB co-expression data indicates the top 3 LHb afferents activated by various stressors are lateral hypothalamus, lateral preoptic areas, and the interpodonticular nucleus. And among those afferents, LH, lateral hypothalamus, were the most activated. So to confirm this data, we then injected the GCAM6 virus in the top 5 activated brain regions. And we implanted an optic fiber either above those brain regions or LHb to record calcium signals from either neuronal soma or their terminals within LHb respectively. By parallel performing these experiments, we observed that all selected upstream brain regions showed large calcium responses to acute restraint stress at the soma level. But intriguingly, at the level of their terminals onto LHb, only LH to LHb axons exhibit an overwhelmingly increased calcium activity after presentation of restraint stress. And similar results were also found with retrosoma fiber photometry recording in restraint stress, tail suspension, foot shock, and social defeat stress. The top 3 activated brain regions were LH, EP, and LPO, while LH exhibits an overwhelmingly large calcium response to all stressors, and this is consistent with our CFOS and CTB experiments. So taken together, we identified LH as the most physiological relevant upstream brain regions that convey stress information to LHb, and we speculated, although multiple LHb upstream brain regions respond to the stressors, there are only a few neurons convey stress information onto LHb, and those axons are mostly likely located within the LH to LHb pathways. And we then focused on LH to LHb pathway and asked if LH to LHb synaptic activity is necessarily required by depression onset. To address this, we used a classical depression model, the chronic restraint stress model, in which we restrained mice in a conical tube 2 hours per day and for consecutive 14 days. After 14 days, we tested the mice with forced swimming test and sucrose preference test, which are 2 well-known experiments that model despair and anhedonia phenotypes. We confirmed that this paradigm could induce severe depression-like phenotypes. And we then silenced the LH to LHb pathway by a chemogenetic approach. We injected synapsin HM4D in LH and implanted bilateral cannulae above the LHb. So every day before restraint stress, we locally infused the DCZ to chemogenetically silence LH to LHb activity. And by daily silencing the LH to LHb activity, we successfully prevented the depression phenotypes induced by 14 days restraint stress. This data indicates the LH to LHb synaptic activity is necessarily required by chronic restraint stress induced depression onset. We then asked how LH to LHb involved in stress information processing. Previous studies have revealed that negative emotions can be artificially induced by instant, strong optogenetic activation of various LHb afferents. However, these investigations are not physiological because they didn't mimic the real stress coding. Here, we found LH is the most physiological relevant LHb inputs under stress. It is important to first decode the physiological activity pattern of LH in response to stress. So by single unit recording, we found restraint stress elevated the basal firing rate. And then we noticed a very interesting phenomenon. When we analyzed the cluster of spikes which exhibit a frequency higher than 10 Hertz, we found restraint stress shifted the intra-cluster frequency from 20 Hertz to 40 Hertz. And this rule is applied equally to various stressors, including tail suspension, foot shock, and social defeat stress, indicating this 40 Hertz neuronal coding may represent stress coding universally. So to test the physiological meaning of this stress coding, we therefore injected the O-chief in LH and performed slice recording in LHb. So all synaptic responses were triggered by a minimal photostimulation, which reliably evoke action potentials in O-chief expression neurons of corresponding LHb upstream brain regions in each mouse. And surprisingly, by photostimulating the LH to LHb terminals with pulsed 40 Hertz chains, we observed a summation of EPSPs at LH to LHb synapses. So which you can clearly see from this zoomed in picture. The decay time of each EPSP is around 40 milliseconds. Therefore, 40 Hertz pulsed stimulation triggers 4 superimposed EPSPs. And this allows sufficient calcium influx, which drives an eruption of LHb burst. And notably, either too high or too low frequency was not able to trigger EPSP burst pairings. For example, when we lowered the frequency to 20 Hertz, which reflect a basal firing rate of LH, we found it only triggers 4 separate EPSPs. And when we increased the frequency to 100 Hertz, we cannot generate a slow rising plateau, thus fail to generate enough calcium to trigger burst. We then compared all LHb upstream brain regions. We found burst induction fidelity is highest in LH to LHb pathway, and this is consistent with our upstream mapping result. So now we have identified the most physiological relevant LHb upstream and its activity pattern in response to stress. Let's back to the original question: how LH to LHb synapses process stress information? So our previous work has discovered intrinsic LHb burst coding the depression-like state, and this burst depends on activation of T-VSCC and NMDAR. Using MPHR to drive a hyperpolarization-induced rebound burst in LHb neuron itself, which is artificially mimicking the intrinsic LHb burst, we successfully induced aversion and depression-like phenotypes, but in real time. However, in the present study, we found stress activates LH, which in turn drive synaptic burst in LHb. So what is the molecular basis of this synaptic-driven burst? Is this burst carrying the same mission during aversion and depression? Our pharmacological experiment in slice recording demonstrated, similar as intrinsic burst, this synaptic-driven burst also depends on T-VSCC and NMDAR activation. We then tested if this synaptic-driven burst can also induce real-time aversion and depression. To test this, we first opto-tagged the LH to LHb synapses in vivo, and we found photostimulation with 40 Hertz at LH to LHb terminals reliably induced LHb burst in freely moving mice. So here we photostimulated at the LH to LHb terminals with 40 Hertz chains, as we clearly see in the left video, and this synaptic-driven burst elicits immediate immobility in fast swimming test, which indicates despair phenotype. We also observed real-time aversion without change of locomotion. However, 3 days after the withdrawal of short photostimulation, the despair-like phenotype is completely reversed to normal. And this phenomenon is of particular interest. We have demonstrated that LH-LHb EPSP burst pairings encode stress. During chronic stress, this EPSP burst pairings is supposed to be repeated again and again. So does it induce any persistent change, sort of plasticity? So let's look at these EPSP burst pairings. Does it remind you something? The 4 EPSPs represent the presynaptic events, and the calcium influx-driven eruption of burst present a postsynaptic event. The time window between the 2 events are very close. Yes, this reminds us the Herb's postulate. Cell fires together, wire together. So this suggests that this stress coding, the coincident EPSP burst pairings, may lead to persistent plasticity that causes hyperactivity in LHb and induced depression. So we apply this EPSP burst pairing specifically at LH-LHb pathway by optogenetic approach. We reliably elicit the LTP. And this phenomenon is very exciting, since previous studies found LTP is extremely hard to be induced in LHb. And LTD is dominant. So why? Imagine, LHb hyperactivity is a marker of depression status. If it is too easy to be potentiated, then all of us will be depressed. Thus, a healthy individual must confine the occurrences and robustness of LTP in the LHb to cope with stress. However, pathological hyperactivity of LHb is a widespread phenomenon under depression-like state. So there must be a way to potentiate most LHb neurons during stress, but very difficult. So here we have found it, this EPSP burst pairing induced potentiation. And this LTP requires both pre-EPSPs and post-burst coexistence. So in support of this view, EPSPs represent the presynaptic events, or burst represent postsynaptic events alone failed to elicit any LTP. So now we have a short summary for the first part. LH fires at 40 Hz under stress, which drives EPSP summation and a subsequent bursting in LHb. Apply EPSP burst pairings at LH to LHb synapses, we could induce LTP. Based on this information, we speculated stress drives LTP, and this LTP should take place specifically at LH to LHb synapses. So to test this hypothesis, we performed an occlusion experiment. So what is an occlusion experiment? We know that all synapse has a plasticity capacity. If stress-induced depression onset requires such an LTP-like process, the animals experience chronic stress will possibly show decreased plasticity capacity. In another word, a saturated plasticity. So we apply the EPSP burst pairings protocols at LH to LHb pathway in already stressed mice. So as expected, we failed to induce any maintained plasticity, indicating stress leads to LH to LHb synaptic potentiation, which caused a ceiling effect in LH to LHb synapses. In addition, we compared pair-pulse ratio between naive and chronic restraint stress in variable afferents onto LHb. As we show here, PPR was significantly decreased at LH to LHb synapses, indicating an enhanced presynaptic release probability selectively occurred at LH to LHb synapses. Similar phenomenon was also observed with chronic social defeat stress. Okay, so in reviews of Martin and Maurice, they proposed plasticity is induced during memory formation, and is both necessarily and sufficiently required for the memory mediated by the brain areas in which that plasticity is observed. So this means if there's a LTP during stress at LH to LHb synapses, we would be able to create an artificial stress experience by inducing LH to LHb LTP. So we optogenetically applied this stress coding, the 40 Hz EPSP burst pairing protocol at the LH to LHb terminals to mimic the 14 days chronic mild stress. And this is a mouse who received daily photostimulation in home cage without any other additional stressors. And after 14 days, we found 40 Hz EPSP burst pairing protocol induced robust depression-like phenotypes, including this increased immobility in forced swimming test and anhedonia in sucrose preference test. And we also observed enhanced EPSCs in achieved tagged neurons. Meanwhile, 20 Hz, which induced only 4 EPSPs, failed to elicit both depression and LTP. So taken together, our result identified LH as the most physiological relevant input onto LHb in response to stress. It fires with 40 Hz cluster of spikes, which triggers EPSP burst pairings at the LHb neuron. By repeating this stress coding at LH to LHb synapses, we were able to mimic chronic stress and induce both behavioral phenotypes and LTP in completely naive mice. And finally, back to our goal of this project. We really hope we can continue and discover more in this direction, sort of targeting the LH to LHb plasticity to prevent stress. We hope our work will make a big contribution in clarifying the neuromechanism of stress-induced depression. And bit by bit, we hope we can tame the black dog eventually. And this work was mainly done by Yihui and Guo Chen. Yihui is a super talented and passionate PhD student. She has magic hands and she's performing the most perfect surgery I've ever seen. She's also working hard, easy to communicate, and self-motivated. Chen was initially my technician, and she's very smart and plastic, and she gives very good presentations, and I witnessed her super fast grown up. Now she's already a PhD student in my lab. And besides, I would like to thank all the members in my lab, and my great thanks to my postdoc supervisor, Hailan Hu, and all other collaborators, colleagues, friends who give me support and help. And thank you for your attention. And finally, postdoc position is always available and welcome to join us. Let's tame the black dog together. Thank you.
Thanks so much, Yihui, for a great talk. And we have time for questions.
Okay, Yihui, wonderful talk.
Yeah.
So how long does it take to how long does it take to activate the habenula neurons, and how long I mean, for the optogenetic you stimulated them for 2 weeks, but for like a behavioral paradigm, so how long do you activate neurons, and how long does it take for you to get a persistent activation, so to speak?
Actually, we performed 2 set of experiments, and I showed here, we activated the neurons like 14 days and 2 hours per day, but with like half an hour on and half an hour off. But actually, I think it would be a much shorter time to activate, potentiate the habenula neuron, because when we performed a facilitation experiment, we used subthreshold restraint stress 3 days, and then coupled with this photostimulation 3 days, and we were able to facilitate the depression onset. I think maybe like 3 or 4 days is enough to trigger the plasticity, but we would try later, because I think it's very strong protocol.
Okay, thank you.
Thank you.
quick question, Yihui.
Mhm.
Beautiful work. So, so how do you comment that the LH also has connection to the PVT, periventricular thalamus? Do you think so? How is the synaptic connection, or in subpopulation, LH might play a different role in different connections?
I think yes, LH has a very powerful role in regulating a lot of behaviors.
Yeah.
And one of my think is that because like with our CTB and C-force co-expression, it's although it's like overwhelmingly larger, greater than other afferents, but it's just like 20% of the total neuron. So I think first of all, and maybe there's only a like a proportion of neurons in LH is activated by the stress and convey the stress information onto LHb. And also, we have also performed cell type specific experiment like VGLU-T-Cre and GET2-Cre neurons. And we found that only manipulation on VGLU-T neurons can have the same effect on the behavioral phenotype and long-term plasticity. And also because LH, there's a lot of orexin neurons, and I think maybe there's some kind of regulation, and also I think maybe there's feedback loops from other circuits like neuromodulators, and then the plasticity will have a feedback mechanism to like be overcome the plasticity occlusion, the ceiling effect, or something else.
Right, sure. Okay, you got some questions, Jenny Bar.
Lee Joe, do you want to ask your question?
Mm-hmm.
Yes, it's a nice talk, I think, and I think one of the question another person also asked is why 40 hertz? Is there any specific meaning in the like for the what's so unique about 40 hertz? So this is I think it's a general question.
You mean the 40 hertz from Yihui Cai's work, or like?
Yeah, I think it's from your results. You are showing maybe 10 hertz or even 100 hertz didn't work, but 40 hertz. Right? Or my understanding maybe something wrong. Yeah.
Yeah. So I would go back to this slide. So you can see these 40 hertz triggers 4 superimposed EPSPs. And the tricks of this burst eruption is that you can only trigger 4 consecutive EPSPs, and which force in the first EPSPs the decay time, force within the decay time. And then the 4 EPSP will drive something like a slowly rising plateau of calcium influx. And this is very important because let's see the 20 hertz and 100 hertz. And you can see that if you lower the frequency to 20 hertz, and each EPSP is separated with others. And so they can't generate a slowly rising plateau potential. And thus there's not enough calcium influx. And then the 100 hertz, because it's too fast, it's like a very intense, strong stimulation, like in once. And so it can't generate a slowly enough rising plateau, and then still not enough calcium influx. And the interesting thing is that the LHb is a region that's very specific because it can fire with burst firings, depends on NMDAR and T-VSCC. And we try to perform when we inject the OCHIF in various upstreams, and we also try to patch the neurons from other downstream of those afferents, of those upstream brain regions of LHb. And we found that only hypothalamus can have this only only thalamus can have this the similar burst eruptions when we injected this 40 hertz stimulations. So I think the LHb has this very coincident features. Like first, LH, the stress coding of LH is 40 hertz, and it can drive the very slow slow calcium influx. And then LHb, it has the feature that they can have intrinsic burst firing, depends on NMDAR and T-VSCC. So together, and this drives this 40 hertz EPSP burst pairings.
Thank you. Very clear. Nice job. Thank you.
Thank you.
Kaiwan, do you want to ask your question?
Yes. Hi, Yihui. Very nice talk. So I'm wondering whether you have tried to induce like a depotentiation or such the kind of a LTD activity in this synapse in the stressed animals, and see whether, because you've been showing great evidences to show that the LTP is well correlated with in the stress, and LTP is sufficient to induce the stress behavior, but whether have you tried to reverse this LTP and see whether the stress behavior can be eliminated? Because I remember.
Super good question. Actually, it's our ongoing project, and we want to target LH2 LHb LTP to prevent the stress coding, and we have already some optimizing and I think we will soon organize a paper, and we will show later.
Yeah, that's very cool. And also at the very beginning, you've shown that the CBT CTB injection actually shows there's multiple inputs to lateral habenula. So how about the other inputs to this to LHb? So I'm just curious, because LHb in the stress animal, the spontaneous firing shift to the 40 hertz or higher frequencies. So this bursting activity won't couple with the other inputs to strengthen the synapses, the other synapses as well?
Okay, so we have like 3 criteria mapping, unbiased mapping in the beginning, and I didn't show the data. Here, let's check. So first, we have this electrophysiological mapping, and we found that the different various upstreams, they have different functional connectivity with LHb, and with LH and EP with the highest connectivity. And like LS, it's also activated by the stressor, but with a very little connections with lateral habenula. And also, this is the first evidence. And then we also have fiber photometry with various stressors, and we can see that LH, this is retrosoma fiber photometry activity. And so let's see, this is LH in three stressors, under 3 stressors, and the calcium activity is the largest, like comparing with other upstream brain regions. And this is EP, and this is LPO, and this is LS, and this is S. And so we would not exclude the possibility that the other upstream brain regions is involved, but we would just say that LH is the most physiological relevant upstream brain regions, and it's like universally coding all the stressors, and like in all the stressors, under all the stressors, it fires with 40 hertz. And also we have some data, like also because we have this EP, the connectivity of EP with LHb is also very high, but we noticed that EP, it fires with 80 hertz cluster of spikes, in which we demonstrated that 80 hertz, like very high frequency stimulations, it won't trigger EPSP burst pairings. So maybe LH, this for LH, this 40 hertz unique firing pattern, it encodes very specifically the stress and for the long-term, like persistent plasticity on this aspect. Yes.
I see. Very nice. Yeah, very clear evidence. Thank you.
Thank you.
Okay, Yihui, great talk. Congratulations on the new study. And Zilong, you're up for the next speaker.
Okay, thank you.
Great.
I just stopped the sharing, right?
Sure.
Yes.
Great. Thank you, Yihui, for the beautiful talk in her young lab. I'm happy to first met Yihui like when he just started postdoc in Highlights lab. Now Yihui has own group, have creative group. Now I'm gonna introduce another outstanding young neuroscientist in China, who is Dr. Jianqin. So Jianqin has a very long-standing interest about oligodendrocytes. It's an interesting study his career in PhD and postdoc. So he had a bachelor degree and PhD training in the third military medical university. After that, he also went to France. He has a postdoc in the College of France. After that, he went to UCSF, working with Stephen Fancy, working on the molecular mechanism underlying oligodendrocyte, has a series of outstanding discovery. A paper just published last year, 2021, lasting for over 5 years. So in 2017, so Jianqin started his own lab in China, and in his young lab, he already has a series of interesting discovery. Including one he will talk about today. So he will talk about the oligodendrocyte is an interesting ring finger protein RNF43 has an interesting role in injury responses. So welcome, Jianqin. Take away. Yeah, we can see your slide. Go ahead. Yeah, great.
Yeah. Thank you, Professor Cho, for your introduction. Good morning, everyone, and of course, good afternoon, depends where you are. In the very beginning, I want to thank Professor Cho and Aaron Gittler for providing us this platform. I think it's a great opportunity to share our research work online during this COVID-19 age. My talk today is oligodendroglial ring finger protein R43 is an essential injury-specific regulator of remyelination. So this is a cooperative project with Professor Stephen Fancy in UCSF in United States. Okay, the mammal brain contains two parts, gray matter and also the white matter, which compressed mainly by the myelinated axons. In this scanning program, we can see the red part outside the neuronal axon is a myelin sheet. In the electron microscopic imaging, we can see those ring-shaped myelin sheets from transverse section. So during development, the myelination process can only be executed by oligodendrocyte precursor cells, OPC, after they differentiate into the mature oligodendrocyte, OLs. Then they can myelinate the axons, facilitate the neuronal impulse conduction, and provide the axons with the nutrition. Therefore, the integrity of the myelin is critical for normal functioning of central nervous system. However, this myelin structure are easily got damaged in different types of diseases, such as the typical demyelination disease, multiple sclerosis, the newborn neurological disease, white matter injuries, and some neurodegeneration disease, such as AD, and the mental disorders, such as schizophrenia. So in all those pathological conditions, OPCs are supposed to differentiate into mature oligodendrocyte and then remyelinate the axons, like what they do in the development. So people can believe that the OPCs, they just need re-migrate into the lesion areas, then they can differentiate into mature oligodendrocyte and remyelinate axons. So people think maybe the remyelination is a rerunning of the myelination. So that means the remyelination may share the same regulator mechanism with the myelination. In fact, however, the remyelination, especially in human diseases, is usually failed, leading to the axonal degeneration and neuron cell death, and causing even more severe neurological symptoms. So we wonder why the remyelination and myelination are different. Why the remyelination is so hard in diseases. To address that, we asked whether the remyelination is really the same as the myelination in development. Are there any regulators that operate specifically in those injuries? So this is our first question in this study. Before we dive into full research of the specific regulator, among thousands of molecules, reviewed the previous study and noticed that the demyelination-related diseases shares a similar overactivated Wnt pathway with the colon cancer. Because the Wnt pathway has already been considered as a checkpoint of OPC differentiation and myelination, we got some hints from, so we think we can maybe get some hints from the colon cancer research, and also we already get those very interesting candidates which also linked to Wnt pathway. Among those candidates, the ring finger protein R43 caught our eyes. The R43 was first identified in the intestinal crypt stem cells, along with its highly related E3 ubiquitin ligase like ZNRF3. They both contain extracellular domain, transmembrane domain, and cytoplasmic ring domain. They are not only the Wnt pathway target genes, but also negatively regulate the Wnt activation by ubiquitinating the Wnt receptors, in particularly the physioreceptors. So whether the R43 is the specific regulator we're looking for, we first assessed the expression pattern of R43 in human white matter injury tissues and demyelination mouse models. I hope you can appreciate that those R43 positive bipolar morphological cells only appeared in human white matter lesioned areas, but were not even detectable in those non-lesioned areas. And also, those R43 positive cells were only co-labeled by PDGF-alpha receptor, which is usually used for OPC stain. Similarly, in this lecithin-induced demyelination mouse model, we also can see the increased expression of R43 in OPCs after the injury. So in order to assess the upregulation of R43, is it only happened in injuries? We tested the R43 expression level in development models. And we found that a transient expression of R43 oligodendroglial lineage, as shown here, the R43 was only co-labeled by the OPC marker PDGF-alpha receptor, but not with those mature oligodendrocyte markers like the CC1 and the myelin-basic protein MBP, considering the absence of R43 in non-lesion OPCs in adult mice. So our result suggests expression model just like this. The R43 is expressed by OPCs in the developmental stage and downregulated to an undetectable level in adult stage and re-upregulated again after the white matter injury occurs. Actually, to further confirm whether the R43 is only expressed by the developmental OPC and OPCs in injury, we also adopted another 3 reporter mouse lines like the LGH11GLP, 63CR1GLP, and 7GLP. In all those developmental and demyelination models, R43 was only detected in OPCs, but not in other central nervous system cell types. So our next question is, what causes this upregulation of R43 in injuries? As I mentioned previously, the R43 is a Wnt target gene. That means the R43 maybe could be like upregulated by the high Wnt tone in lesion areas, since so many other central nervous system cell type can release the Wnt ligand into a microenvironment after injuries. So we conditionally looked out this obligate Wnt pathway repressor APC, especially in OPCs. And our results show that we successfully triggered this upregulation of R43 proteins and MR levels. More convincingly, when we treat the Y-type OPCs with a Wnt ligand in different concentrations, we found that the R43 expression level were increased accordingly with the increase of a Wnt ligand concentration. As shown here, we use the Axon2 and the Norton as the Wnt pathway indicators. And the R43 level just goes up with those two Wnt indicator levels. Of note, this Wnt activation mediates the upregulation of R43 cannot be copied by the ZNRF3. The highly related E3 ubiquitin ligands, as shown here, the ZNRF3 was not upregulated either in vivo or in vitro models. So taken together, I hope I have shown that the R43 but not the ZNRF3 is a specifically expressed factor in OPCs after injury. So our following question is, what is the role of those upregulated R43 in OPCs? Despite the transient expression of R43 during development, our in situ hybridization staining for the mature oligodendrocyte marker MAG-MAG show that those RNF43 single knockout, the ZNRF3 single knockout, or the double knockout could not significantly cause the deficit of the differentiation at postnatal 10 days, the very fast myelinated age of the mouse, implying that the function of both R43 and ZNRF3 are dispensable for timely development. However, when we use this mice to introduce demyelination models, R43 single knockout and double knockout, they had a very similar phenotype on the OPC differentiation, but the ZNRF3 single knockout, they had no effect at all. Moreover, in those EM images, we can see the knockout mice have extremely lower number of the myelinated axons. Even when we extend our analyzed time point to 50 days after the lesioning, we still can detect this permanent reduce of myelin thickness as shown here. So that means the R43 is important, is essential for the remyelination. So what give us a talk another time, someone pointed out that we use the P10 mice for the development experiment and the adult mice for the regeneration experiment. Whether this mass age matters for the phenotype differences in myelination and remyelination. To address that, we also adopted another neonatal chronic hypoxia mouse model. In this experiment, we can see that under the hypoxia condition, the wild-type mice have a impaired oligodendroglial differentiation, but still so, we got this amount of OPCs can differentiate into mature oligodendrocyte. However, when we compared the differentiation between wild-type and the knockout mice, we can see there were extremely rare mature oligodendrocyte in the knockout mice. So by using this evidence, I want to show that the mouse age doesn't matter for those phenotype differences in myelination and remyelination, but the stage of development or injury matters. Very similar with the lecithin-induced demyelination in adult mice. The R43 single knockout and double knockout in the young mice had the same phenotype of the OPC differentiation, but the ZNRF3 had no effect at all. So taken together in this part, we demonstrated that R43 is required for the remyelination in injuries, but dispensable for myelination in the timely development. So what is the underlying mechanism? Why R43 is so important and required for the remyelination? When we treat the wild type and knockout OPCs with a different concentration of Wnt ligand, we notice that although the high Wnt tone environment inhibit both wild type and knockout OPCs going through the differentiation process, but when we compare the wild type and knockout OPCs, we found that those wild type OPCs, they had stronger strains to resist Wnt pathway activation and got some cells differentiated, like here. However, those knockout OPCs, they were more sensitive to Wnt pathway activation. There were much less OPCs can differentiate. Conversely, the gain of the R43 function partly repressed the Wnt pathway activation and promoted the cell differentiation as shown by the increased MBP-MR levels. So our data show that the R43 controls OPC differentiation by selectively regulating the Wnt pathway activation. But how does the R43 resist the Wnt pathway activation? Previous studies have reported that R43 can target the surface-expressed Wnt receptors, especially the physioreceptors, to lysosomes for degradation. But which physioreceptors is regulated specifically by the R43 in OPCs? Since the OPCs can express more than 10 types of physioreceptors according to our sequencing data. So by collecting the membrane protein from both wild type and the knockout OPCs, we found that only physio-1 receptor was increased in knockout OPCs. Our core-IP experiment also confirms that the R43 and physio-1 receptor physically interacted. And our core transfection experiment further confirms that those surface-expressed physio-1 could be relocalized into the cytoplasm by co-transfecting with the R43. We even found the increased the physio-1 expression level in the knockout mice after the demyelination. So our result demonstrates that the action of the R43 downregulated physio-1 surface level might be responsible for the role of R43 in resisting Wnt pathway activation and promoting OPC differentiation in the lesioned areas. Okay, our final question will be whether our findings can be used as a target to promote remyelination in injuries. The UM206 is a synthetic peptide derived from regions of high homology between Wnt ligand, such as Wnt3A and Wnt5A, that acts to interfere with the interactions between Wnt ligand and the physio-1/2 receptors. In a simple word, the UM206 is a selective physio-1/2 antagonist. So when we used those UM206 in our cell culture, we found that those OPCs in a high wind tone environment, the UM206 could significantly reverse the Wnt pathway activation and enhance the MPP expression. Like shown in this group, there are more MPP positive red cells here. So our results indicate that targeting the physio-1 receptor can help those OPCs in a high wind tone environment to resist their Wnt pathway activation and got more cells to differentiate and further remyelination. Then we want to bring our study to our next level to answer whether this approach can really be used to promote the remyelination in injuries. We test the effect of UM206 via ex vivo cerebral slice culture induced demyelination model. In this experiment, we can see those brain slides from the knockout mice showed no remyelination at all. But when we treated those slides with the UM206, the remyelinated axons, as shown by the co-labeling of MBP and the neurofilament 200, increased strikingly. Furthermore, when we delivered this drug directly into the lesion areas in the brain by using the osmotic millipump, we can detect greater number of the mature oligodendrocyte, the more myelinated axons, and the thicker myelin sheaths. So we suggest that the approach of targeting the R43 regulated physio-1 receptor could be a potential therapeutic strategy for demyelination diseases. And our study was published on Neuro last year, demonstrating those 4 main points. We showed there is a difference between the myelination and the remyelination, identified as injury-specific regulator, the R43, and figured out its mechanism and implied its potential roles in the treatment. Last, I want to thank all those people who contributed to this project, especially Dr. Stephen Fancy, who is a leading corresponding author in this project, and the first and also co-responding author in this paper. And last, I want to thank all of you for your attention and appreciate for all your questions. Thanks.
Great. Thank you, Jianqin, for a beautiful talk. Yes. Now it's open for questions. Yeah, Hongyan, please.
Very nice talk. So I'm wondering how exactly is the RNF43 expression upregulated upon injury? Does it going through the upregulation of the Wnt ligand? And also, well, which type of cell produce the Wnt ligand upon injury?
I think R43 is definitely regulated by the Wnt ligand, but which cell type can release the Wnt ligand in the injury, we cannot identify that. But according to previous studies, there are so many cell types can release the Wnt ligand. Even OPCs themselves, they can produce the Wnt 7A, 7B. That definitely will outcream in an outcream manner to regulate themselves. Yeah, that would be my answer.
Great. Hi, Jianqin. I'm curious about so we know the demyelination actually is also happen in some other neurodegenerative disorders, right? So have you seen the activation of RNF43 in other neurodegeneration disorders?
Like, so far we
Alzheimer's. So I don't know.
So far we didn't check another like human tissues with a degeneration phenotype, but we definitely see some R43 positive cells in the schizophrenia, which is also demyelination-related diseases. Yeah, I think the R43 just like regulated by the Wnt pathway activation. So no matter what situation, what state, as long as the Wnt pathway is upregulated, the R43 could be expressed.
Okay, yeah, just some more demyelination disorders, right? In my besides MS, so I think maybe
definitely besides MS. We also showed some staining not here because of the limited time. We also found in the HIE tissue, it's a newborn injury. They also have a upregulated R43 in OPCs.
Interesting, great. All right, is more questions? All right, and Aaron, do I have any more questions?
No questions for me. Great talk, Jianqin.
Thanks. Maybe
okay.
I'm
maybe I'm
looking for the
talk. Thank you.
Thank you very much.
Yeah, thank you for your great talks as well. Great. See you all next week.
Yeah, see you next time. Bye.
Bye. Bye.
Thanks, Yihui. Thanks, Jianqin. Good to see you.